so much for this intro. It's always the most nerve-wracking to get to talk on your home turf, especially when about a third of the audience is from my own turf. But I'm excited about that. At least I get to hear the story from my own mouth. They probably have already noticed that this picture is way out of date. We need a new group photos. Please help schedule that. For those of you who don't know that, here's the group. They're great. And I am really excited today to tell you about one aspect of our work that's been going on. Actually, the entire time the lab has been around, even before that. I'll give some of the historical context as we go through. Before I start, I want to give a really quick group overview. As Sangeeta mentioned, I'm in the Department of Chemistry. I like to design and make new molecules. The targets I'm interested in making are ones that can solve problems. That would be the goal. And I would say while we are, we don't discriminate in terms of the size of the molecule. We make small molecules, we make big molecules, we make materials. And we don't really care about what synthetic methods or strategies we use to make those. However, we can think about our research over the years and break it down roughly into 3 different application areas where we design molecules to solve problems. And those are shown here. So I'd say about a third of the group today works on designing new molecules for energy storage. I'm not going to talk about this, but if you're interested, check it out. About another third, I would say, is interested in making, designing new molecules to make sustainable material, plastic and thermoset materials. I'm not going to talk about that at all today. But last but not least, another, say, roughly a third of the group is designing new molecules to address problems in healthcare. And I would say, I think each of these energy storage, sustainable, and healthcare are arguably the 3 most important things people can work on. So I'm happy to be in that space, and I'm really proud of my group for making contributions in each of these areas. Okay, so we're going to jump right into the last one, which is the focus of today. And I'll start off by introducing a concept that I don't think anybody in this audience needs to be introduced to, the idea of the therapeutic window. Well, again, I think you'll know what this is. I just want to make one general statement, which is that regardless of what type of drug you're developing, what the mechanism is, what the modality is, doesn't matter. I think it's generally always good to have a wider therapeutic window. I can't think of any case where you say, no, I want to therapeutic. All right, and so throughout the entire history of drug development, one could argue that from a certain perspective, it's literally just the search for molecules with wider therapeutic windows, regardless of disease context. And many new revolutions in drug development are simply just a new way to approach expanding therapy. And so I'm going to review and highlight 2 of those ways, or maybe others, but 2 of them that are most relevant to this topic. Again, the first one is the advent of what are known as molecularly targeted therapeutics. These are generally small molecules designed by medicinal chemists where modifications to a small molecule structure are made to optimize the binding of a small molecule to a specific biomolecule implicated in a disease pathway where the disease cell, in the context of today's talk, we'll talk about cancer, so I'll just say cancer, or we can keep it more general than that. So, for example, when we want to design a drug that is selected for a biomolecule that is necessary for cancer cell survival, but is not important or less important for healthy cell survival. And that's a way to build a therapeutic window, right? So I would say these drugs operate through a mechanism of action-based therapeutic window, right? And this, after the advent of the early chemotherapies and molecular targeted drugs came on board, and it led to dozens and dozens of compounds that are used in the clinic today. They still haven't solved all the world problems, obviously, and that largely boils down to the fact that it's rather rare for there to be a mechanism of action that's 100% selective in a cancer cell and not present anywhere else in the body. So these drugs can still have off-site, sometimes off-target toxicities that lead to relatively narrow therapeutic windows. Now, this is based on programming this mechanism of action to a small molecule structure. Another way to think about improving the therapeutic window is to bring drugs directly to targets, and that's maybe most relevant for the nanomedicine audience. And I would argue perhaps one of the best examples of a nanomedicine is antibody drug conjugate. Antibody drug conjugates, probably know, involve a monoclonal antibody designed to target a receptor of cell, ideally one that is not present anywhere else in the body, but more often than not, actually simply overexpressed down the cancer cells and on healthy cells. And to that monoclonal antibody, one conjugates payload molecules that, once the antibody binds to the target, taking the cell, releases those payloads. You kill that cell, and hopefully some nearby cell as well. Now here, antibody conjugates are been very successful, especially in just the past few years with some work. Maybe most notable from the Conjugates and HER2, which I'll bring up again later in the talk. But ADCs, as remarkable as they are, they still have significant drawbacks. One of those is, people often think of these as antibodies like a homing device to a target cell. But in fact, an antibody doesn't have a mind of its own. Like it doesn't say, oh, I'm over here, the tumor's over there, I'm going to swim to that. Right? It simply distributes around the body via passive biodistribution. And when it happens to find a target cell, it can bind and kill that cell very efficiently. But it's now quite well understood that less than 1% of injected dose of ADC actually ever find a target cell, which means you're inherently exposing off-target tissues to these highly potent payloads. Okay? And that leads to their therapeutic window. Even the best ADCs in the market have significant known toxicities. And so, from a very naive perspective, one idea we had was, why not combine these two paradigms? Why not employ molecularly targeted small molecules in the context of antibody-drug conjugates? Very simply, thinking this through, what this would mean is that for every antibody-drug conjugate molecule that arrives at its target cell where the antibody binds, it could also bring to that target cell a small molecule payload that is more inherently selective for killing that cell based on its molecular mechanism of action. And for every molecule that doesn't arrive at its target cell, it brings a payload that is inherently less toxic to that other, whatever that other cell is. So that it's very simple. It's essentially trying to multiply the effects of these two ways of expanding the therapeutic. Okay. Now it also seems very obvious. So the question of why don't we do that? And the answer has to do with a key limitation of antibody-drug conjugates. And that goes back to what was captured in that cartoon. You may notice there were only 8 little red circles attached to the antibody. That has to do that we define a number of drugs attached to antibody using a term called the drug antibody ratio star. Figures star a lot. And an antibody is a pretty big molecule. They're like 150 kilodaltons, roughly 10 by 15 nanometers in size or so. And if you can only conjugate 8 drugs, you need those drugs to be extremely potent compounds. Right? And that's turned out to have been shown to be true throughout the decades of research in this area. So this is a figure from a review article actually written by the scientists at Daiichi Sankyo, where they classified different payloads into the ones that have been successful in ADCs from this line to the left to the ones that haven't. And this x-axis here is just payload potency. And basically what it shows is for drugs to be successful in an ADC, they need to have essentially sub-nanomolar potency. And it's actually really hard to make drugs that have sub-nanomolar potency. There aren't that many of them, and it's why even though there are maybe a few dozen ADCs in the clinic today, they all use a small set of payloads that like 2 or 3 mechanisms of action. And so the question is, how can you expand that into inherently less potent for these molecularly targeted drugs, where we might have potencies maybe single-digit nanomolar, maybe 10 nanomolar, maybe 100 nanomolar. And in case you don't believe me, here's a long quote from this review. I'm not going to read it all here, but the last line is, thus, the discovery of novel toxic drugs or ADC payloads with different mechanisms of action is desirable. And I do think that in the ADC space, there's basically like 2 major thrusts of research. One of them is designing and developing new antibodies, finding new targets, and that's awesome area. We don't do that so much in my lab. The other is to try to enable new payloads and new payload mechanisms. That's where I would focus. All right. So going back to this cartoon, we can have, it used to be 4 species to be the max, but now with the HER2, you can say 8. So clearly you can increase the dog, but now it's kind of thought that 8 drugs is the max. All right. And so how would you go to using a drug that's 10 times less potent, 100 times less potent? Well, you could say, well, I'm going to dose 10 times more ADC, get way more. And that might work in some cases, but the challenge there would potentially be that you'll start to saturate all the receptors and antibodies are meant to bind you. And anything above that in terms of the dose, you're just going to be essentially passive delivery that won't actually give an additional. So if we say at the same antibody loading, how do we use the drug that's 10 times less potent? We need to increase the DAR by 5%. 100 times less potent. Increase the DAR by 100. That seems straightforward. However, it's not so trivial because if you just take a molecule of antibody and add 10 times more payloads to it, there's a really good chance you're going to disrupt this function. First, by simply binding to sites that block its ability to recognize an antigen, but even more fundamentally, most payloads seem to be rather potent compounds. This will start through disaggregation, the physical properties of the antibody will be ruined. This pathway is not really wide. And again, like I said, 15 years ago, people would say you can't go above 4. Now there's 8, but people really say you can't go back. All right, at least through this type of strategy. So an alternative way that we started to think about was, well, what if instead of attaching 20, 30, 100 drugs directly to the antibody, we decouple the connection points of the antibody and instead attach some chain where each repeat unit of that chain has the payloads and the linkers attached to. And simply attach that chain at only one position on the antibody. Now, this would have the advantage that we could make this chain arbitrarily long and have many drugs and linkers we want without having to make that many connections to the antibody. But it doesn't solve these problems about aggregation of physical properties of drugs, right? Because if these are hydrophobic compounds, you make a long chain of attachment, but it really starts. And in fact, this is not a new concept. Like there's a company called Persona Therapeutics, raised lots and lots of money, a very high profile, for essentially having a polymer chain like this with drugs attached. But I'll note in their case, in between each payload, there's about 10,000 daltons of filler mass that's just hydrophilic polymer that helps keep everything solid. So really the spacing change is extremely long. And at the end of the day, despite having a very high molecular weight polymer, they end up with a DAR of 15. So it's maybe roughly doubling the DAR, but with the addition of adding this giant polymer polymer. So I would say our key innovation in the space is very, very simple, but as I'll tell you, it haven't led to all the work that we've done since, is thinking, well, how do we overcome that problem? And maybe we could do it by adding, in addition to linkers and payloads on each polymer repeat unit, what if we also added some blue hydrophilic chain? A blue hydrophilic chain that's longer than the linkers and the payloads, so it effectively shields them, keeping them soluble, preventing this whole structure from aggregation, regardless of what the linkers or the payloads are. Imagine sort of in the extreme coaxial cable, where along the backbone, this rigid linker, rigid chain, you've got your linkers and your payloads, and you've got this sheath of glue around. Right? And such an architecture, if you imagine it, would kind of look like a bottle brush, something you put a bottle out with. But here, the key distinction being that on each of these repeat units, we don't just have the side chains of the bottle brush, we have the side chains of the linkers and the payload built in. All right. And so we refer to these molecules, so forget the antibody part for now, just this part, as Bottlebrush prodrugs. The prodrugs, because these drugs are inactivated and they're attached and have to be released through some mechanism. And we think that they could unlock novel ADC payloads, sort of allow us to break this limited DAR and get into whole new mechanisms of action. All right. So the first question is, how do you make that? And this is going to get into some history. So turns out, I did my PhD at Columbia University, and at Columbia, just like I did my PhD in many places, there's a requirement around the 4th year called the original research proposal. And we're supposed to write some proposal on some topic that's unrelated to your research, and that's why I was looking for anything drug delivery related that I was interested in it. And so I started thinking about it. And I noticed that the traditional design for polymer drug conjugates, because at the end of the day, that's what that molecular bottle brush is, polymer drug is. They tend to have one of two different designs that both share a feature that they have surface exposed payloads. So what do I mean by that? Well, one example I kind of already gave you, that was some example, but here's another one. So one can make a linear polymer and have pendant conjugates where there's some cleavable linker between the payload and the polymer. Here's an example of one. This is a Taxol polymer drug conjugate. And I've actually drawn this misleadingly because it makes it seem like every repeat unit of this this polymer backbone has Taxol, but it actually doesn't. In fact, it's only sparsely functional heads with Taxol. Again, if you functionalize every repeat of this polymer is not involved in water management. But this is one, I would say this is just one general design trick. Make polymers with drug. The other one is to take branch polymers, using star-shaped polymers, so polymers with four and multiple arms, and conjugate drugs onto the ends of those arms. And here's an example of an SN38 polymer prodrug that was studied in 20. And both of these share this feature that they have surface-exposed payloads, meaning that whatever the identity of these payloads is, that will dominate the physical properties of the overall construct. So now, as a graduate student back in 2006, I saw this paper that came out from a team of researchers at Berkeley, and title has this very sort of, I think, scientists having title, a single dose of doxorubicin functionalized bow tie that cures mice and C26 colon particles. And this was happening to be around the time I was getting my fourth research proposal, and I thought, oh, this looks cool. What is this about? And this idea, so first, what is a bow tie dendrimer? So this is a name coined by the Frichet lab. Jean Frichet for pioneers in dendrimer synthesis. And if you don't know what dendrimers are, they're basically the polymers that are highly branched. Whatever you're eating, there's another branch. Right? And so a bow tie dendrimer is essentially taking two halves of dendrimers, putting them together. So it kind of looks like a bow tie. Here's half of the bow tie, here's half of the bow tie. And what they did in this paper was they made a bow tie where half of the dendrimer has polyethylene oxide chains. And on the other half, there's these hydroxyls that they can attach a drug to, in this case, doxorubicin, which they attach to an acyl hydrosome to a pH cleavable linker. You basically have this construct that like half of it is peg chains, hydrophilic, peg, and the right side is a bunch of conjugated drugs. And so in my sort of grad school cartoon form, I drew this like this, where we've got peg chains, we've got drugs, and while this doesn't solve all of the world's problems, it does kind of address that idea of surface exposed payload. Because at least if these blue strands are long enough, you can imagine that they're just going to kind of shield this hydrophobic core for all these payloads. And so this was certainly inspiring, but it definitely had some challenges. One, from the perspective of conjugation proteins like molecular antibody, this overall construct has spherical symmetry, meaning all these functional groups are essentially the same. It'd be really hard to install just one of them to attach onto it. The other, maybe more important one, is just this is really hard. Right, that synthesis on the previous slide. Dozens and dozens of steps. If you wanted to change any variable, you have to go back to the beginning. Really, really, really hard to make. But if you just kind of like zoom out and squint a little bit, you might be able to imagine that a similar kind of design, similar features could be obtained by, instead of having this dendritic core, making a linear core, and then having these branch points to the drugs, have those be on every repeat of that linear core, and then have the blue strands expanding out, just like on the cartoon I showed you earlier. The Bottlebrush. Two potential advantages of this. One is it has a cylindrical symmetry, at least there's an end group versus a middle, and that means we in principle could attach one functional group to the end for conjugation, something else. I'll show you that later. And then the other is potentially easier synthesis, at least from the perspective of, well, I'll show you later how you actually make them, but this could in principle be made by a single polymerization reaction if the monomer itself already has the payload, the linker, and peg chain. Okay, so now, really going into the history, this is a sketch from my own personal journal from June 13th, 2007. So I was walking around the streets of New York City, I was like, what am I going to do for my proposal? And I had this idea, I want to make this, which has, it says hydrophobic, had a backbone, water-soluble thing. There's an X group here, and it was like it says photocleavable to release X. At the time, I was using light to trigger the release of payloads from these types of structures, which I ended up doing. I'm not going to talk about that at all today. It doesn't need to be photocleavable, but that was the idea. And the way one could maybe make this, I wrote something like this, where there's this norbornene group that is attached to this photocleavable linker with an R. This would be a payload. There's a peg chain. By the way, for folks in my group, yes, I know there's a hydrogen missing here, and it drives me crazy to this day, but I'm not going to go back and play it in my own journal. So then if you go to the next page, you can actually see here, where it says drugs here. So we might polymerize this rewarding using a reaction called ring opening polymerization to make a polymer backbone, something like that, and water soluble. And here's a sketch of the New York, of the Columbus station. So yeah, just get it at. All right. So then figures from that actual 4-year research proposal, just put these into actual ChemDraw forms, and here's one of the cartoons in the whole class. So this is literally something I've been doing for 15 years or so, and it's been awesome to kind of see it come to the point where it is today, which hopefully all right. You can also background the actual. Sure. All right, so I'm skipping a lot in between. Started actually making those molecules towards the end of my PhD time. Continued that in the post-doc came here, but still, I'd say the first generation synthesis suffered. Was not really scalable. I made enough to do what I wanted to do with it, but I was happy to have a former PhD student in the lab, Hung Ran, who was like, I don't want to go through this 6-step hard synthesis anymore. So kind of took a step back, redesigned the molecules, and without going into the details, this is the synthesis that we use today. And to make a long story short, we can get to this key building block, and this is the key building block for the following reasons. It has this exonucleotide constituent that we can polymerize. It's got this head chain here, with those are the blue strands of the carbon, and it has this terminal alkyne here, positioned more closely to the rewarding than the end of this head chain. And this terminal alkyne is the place where we can attach whatever payload and linker we want through copper catalytic alkyne position. And so we can get to this molecule in like 4 overall steps with no chromatography required, 85% total yield on the kilo scale. In fact, this is now big data. 10 to the scale. So it's a really efficient synthesis. And then once we have this molecule, it's quite straightforward to do the click reaction, as I mentioned, with whatever azide we want that contains a cleavable linker and payload. And this is actually nowadays where a lot more our synthesis work goes in. Because this kind of stays the same, but depending on what payload we want to use, we have to develop a way to make a cleavable linker azide prodrug of it on its own. And then we do this click reaction, we make this macromonomer, and then call it a macromonomer again, because it's going to be polymerized with also macromonomer itself. And we treat this with what's called the Krebs third generation initiator ion, and long story short, this polymerization gives us the ability to very tightly control the overall backbone length. So look at this purple chain. And so if we put in, say, 100 of these to one of those, we get an average length of 100, and it's very narrowly distributed. And I just want to mention as well that I don't think there's any other polymerization in the world that can do this. So it's really, really cool that works. In fact, when I first got to Caltech for it, I did not believe a polymerization or something like that. Very happy to be proven wrong, but it's still kind of amazing. Okay, so with that as a basic design, we've now I think worked with, this is out of date, probably over 40 by now, different payloads from chemotherapies to molecularly targeted drugs to immune stimulants to PROTACs, even imaging agents. But white things don't need to be payload like drugs. Imaging handles, done MRI, fluorescence, and various things. And I'm not going to go into all the details. I'll just give you one example from a paper we published last year, where we worked with the molecularly targeted drug called bortezomib, also Velcade, which is the first-in-class proteasome inhibitor that is used as a frontline therapy for multiple myeloma. But we did have a lot of potential for treating many other types of cancers, which I'll touch on a little bit later. But you can see the general design. There's been rewarding. There's a head chain. In this case, we've attached to this cleavable linker a boronic ester that, in the presence of various intracellular signals, lead to the cleavage of this boronic ester, releasing a free boronic acid, which is the active one. Now, I'll briefly show you that thing was shown to work effectively. This is in 2 different mouse models of multiple myeloma. And I should say this work was all done in collaboration with Irving Bobriel at the Dana-Farber Cancer Institute. But first, in a sub-Q mouse model, cell line called KMS11, we saw pretty significant improvement in survival and reduction of the tumor volume. So we actually moved into an orthotopic multiple myeloma model. This is MM1S. This is a bioluminescent cell line. We measure tumor burden. That's circulating tumor cells. And what we were really excited to see is in this very challenging model, where actually bortezomib alone at its maximum tolerable dose essentially confers very little to no survival benefit. We were able to cure 40% of mice. And that was enabled by being able to go to a much higher dose without any toxicity because of the prodrug. So we would get longer circulation, less force release, more control, longer area under the curve, and so on and so forth, and see efficacy. And by the way, this is no antibody. This is just a Bottlebrush program. Okay. Other stuff we could do in this paper that I won't have time to tell you about, but I'll just highlight the chemistry really quickly because I think it's cool. One thing this platform can do that I think you can do uniquely well is make combination therapies of different classes of payloads. And in this paper, for example, in the clinic for multiple myeloma therapy, actually the frontline therapy is a 3-drug combination comprising bortezomib, pomalidomide, the immune stimulant, and dexamethasone. And it turns out that 3-drug combination is not like anyone did some combination index assay, found synergy, that this is the right combination, let's use that. It just kind of happened empirically over time that doctors were like, well, just try to say it doesn't have overlapping toxicity. Then eventually, the frontline treatment can settle on this combination. So in this paper, what we did is we actually did those combinations index assays, and we found a combination of 3 drugs actually synergistic. We found other combinations that are negative. And we built those 3 drugs into single Bottlebrush prodrugs at their synergistic and antagonistic ratio to test the hypothesis that having all the drugs built into one carrier, the synergistic ratio, would outperform the same 3 free drugs, even given a synergistic ratio. So the rationale being that when they're built into one carrier, every time one of those carriers gets to a target cell, it brings the synergistic ratio of those drugs to that cell, versus 3 free drugs, which are completely independent by distribution data. So you might find the ratio in vitro and try to go in vivo. And again, I'm not going to go into the details of any of that, so I don't need antibody stuff. I want to show you maybe my favorite market, which has nothing to do with biology, photosynthesis. But it's just showing that here are bortezomib BP, pomalidomide BP, dexamethasone BP, synergistic ratio for all 3 drugs at a ratio that is not on this one. Synergistic one, antagonistic ratio, and then just the Bottlebrush program by itself with no drug as control. And these are site excluded from chromatography traces, basically showing that they're all identical. These are dynamic light scattering histograms showing that they're all identical. And here's just one cryo-EM image, so we have others and they all look identical. So the take home of this is like we can get different payloads with very different physical properties, different combinations thereof, and make these highly reproducible particles that don't depend on what the payloads are. Okay. So now, size and context, though. And nanomedicine syndrome, we all really appreciate that. People throw around the word nanoparticle left and right. And I just want to remind you that's like 3 orders of magnitude at least of size. And I think talk to pharma companies, and I'm like, how many do you like nanoparticles? Like, how do you do like biologics and therapeutics? We love them. So those are nanoparticles too. ADCs are nanoparticles. I hope you agree. ADCs are nanoparticles. And now there's a wide range. Small molecules, they tend to be a nanometer in size. So I just want to put in context where our BPDs are. So they're as I showed you in the DLS on the previous slide, they're about 10 nanometers, which is on the same size scale as a molecule. This is a range that's actually not trivial to access with a lot of synthetic systems. Like, for example, lipid nanoparticles, very powerful technology, but tend to be more in the next order of magnitude. And that's great for carrying large molecules. Like, I don't really like it. We're not going to do mRNA with these with mRNA bigger nodes. But we think there's a lot of opportunities to actually integrate things. We've been doing that for years. One nice thing about having relatively small particles with an aspect ratio that we can control is that they do go penetrate tumors quite well, even though you can't see this. This is that KMS11. It's a cadence model. I showed the results for a few slides ago, and we've labeled the Bottlebrush project as Site 5, and throughout this tumor, you can see fluorescent tumor again. All right. So now, to the real question, this is a much more recent one. How do we do this? So we've got a molecule that's almost the size of an antibody. We have an antibody. We want to couple those together. And in chemistry, that's a hard thing to do, right? And primarily the reason why one is just essentially concentration, right? The rates of chemical reactions are some rate times concentration, like molecular ones, and we have huge molecules that can only be so concentrated. So we're usually working at very, very low concentration. In addition, we have to deal with steric hindrance, so wherever the functional group is on this, maybe market access. So this is generally a difficult thing to do. Now, one nice feature of reopening this polymerization we use to make these is that at the end of the polymerization, we have this ruthenium, called an alkylity, a ruthenium carbon double bond sitting on the end of the polymer. And we got to get rid of that somehow. Usually, we get rid of it just by adding something called ethyl black ether, we just chop it off away, and there's nothing left to do. But it also presents an opportunity. Again, what if we could chop this off and leave behind some functional group X that could be a reactive chemical bioconjugation? And so we were really inspired by work from Bill Prodrugs at Georgia Tech, who showed a few years ago that indeed polymers made by Roth, if terminated with molecules called enynes, which is what this example is, very, very efficiently lead to the conjugation of whatever is on this enine nitrogen onto the end of that polymer chain, with the extrusion of the ruthenium as this stable sulfur chelate compound. So the way to think about this is the ruthenium adds an alkyne rapidly, that then undergoes an intermolecular reclosure, as shown here. And this does two great things. It attaches whatever red thing you want on the end of the polymer in exactly one position. And it gets rid of the ruthenium. And even better, it gets rid of the ruthenium in a really inert fashion that doesn't keep reacting or causing a because the old way of quenching lump gets you ruthenium, that's not completely dead. Right? And that's the discussion. So Van Loo, who's here in the audience, again, building on this inspiration from Will's group, designed this molecule 1, where on that enine nitrogen is this little peg linker and then this tetrazine. The tetrazines are great substrates for yet another one of the so-called chemistry reactions, or the inverse like conjugate Bottlebrush reaction between tetrazines and trans-cyclopyrene. In fact, this reaction is actually a lot faster than even the best of the copper and trade promoted alkyne reactions, probably the most efficient of the so-called quick reactions that we have. So it's a great way to design these. And what's nice here is with molecule 1, we just make our Bottlebrush prodrug, you dump it in molecule 1, and now you have our Bottlebrush prodrug with one tetrazine on the end of the chain. Then all we have to do is get our hands on an antibody that has the corresponding reaction partner, which we can do either through stochastic functionalization of an antibody through glycine conjugation, or we can do antibody engineering and do sort of the thionad approach where we functionalize the food selectively. That doesn't matter so much, but the point is once we have this and this, we can simply mix them together and put them together. All right? And we call these antibody Bottlebrush conjugates. And now, of course, the drug to antibody ratio in the system just depends on how long it's on. So, show you a few pieces of data to show that we can actually make these. This is gel electrophoresis data showing that compared to trastuzumab, so by the way, for the first study, we focused on trastuzumab, the classic HER2 targeted monoclonal antibody, largely in part because we get our hands on high quality cheap antibody to do the study. So you can see that compared to trastuzumab, when we conjugate on an ABC with a DAR of 60, that means the Bottlebrush has 60 competing units. You can see we get a pretty clean single band, indicative of a one-to-one complement to the antibody to the Bottlebrush. We can go to DAR 120, then up a little bit. All right. This is actually about as far as we have gone because we have seen that at really, really high DAR values, we do start to erode a little bit, but antibody binding, so not dramatically. So this is actually, these are microscale from freezes data. We've also done ELISA. This is basically just measuring antibody binding affinity to the target. And comparing trastuzumab, here we actually have two commercial antibody drug conjugates, TDM1, also called Taxila, the original HER2 targeted ADC before Daiichi came out with HER2, which sort of blew everything else out of the water. But you can see compared to these three, our ABC with either DAR 30 or DAR 60 has very similar binding affinity. At DAR 120, it can drop by about a factor of 4 or 5, and beyond that. But still, I mean, I think it's remarkable that we're able to get over 100 drugs on an antibody with a pretty minimal impact on binding affinity, and I really do believe that that goes back to the fundamental sort of polymer physics of a molecular bottlebrush conjugated to an antibody. It's sort of keeping this compact structure that can't really fold around the antibody and block it, like linear polymers, similar hats and so on. These display, antigen-mediated cell uptake. Again, fluorescence is kind of hard to see in here, but this is looking at cells treated with, and this is BT474, so HER2 positive cell line, exposed to ABC that's just using IgG1 antibody that doesn't target anything. This is a control, and you don't go in the cells. And then if we have the ABC anti-HER2, it goes in the cells very well, and then especially in the water. Okay, and then with those in hand, Jen did a lot of different vivo studies to study these different models. We've actually looked at 6 or 7 different payloads. We've looked at different antibodies. This is just one set of data that's quite striking. In this case, is he made an SN38 for 120. And this is BT474 again, HER2 positive cell line, where we see for the ABC that's targeting HER2, we have complete tumor volume regression, 100% survival. And here are the controls are the ABC conjugated to IgG1. So again, sort of non-targeting ABC, mimic sort of active distribution. And we have the BPD alone, which is also not targeting just passive. And it's good to see that the blue and the green basically overlap. Neither of them are targeting growth. They do confer some survival benefit, and that's just due to the sort of just passive targeting of these small particles. But the antibody brush conjugate actually using trastuzumab by far at these doses. All right. Now I want to go back to the original motivation, which was around molecular targeted therapeutics. In the previous example, this used SN38. SN38 is sort of right on the borderline of usable in a ADC. In fact, it is a payload for a drug called Trodelvy, which is a HER2 target. Is that my phone? Very interesting. Sorry. All right. Okay, so SN38. I mean, it's a viable ADC drug, but let's go back to bortezomib, that proteasome inhibitor. It turns out, I'm not going to go through all of that. Proteasome inhibition, as I said earlier, these drugs are used as frontline therapy in multiple myeloma, which is a liquid blood use. But back when it was being developed in the 90s, it was actually found to have single-digit potency, single-digit animal potency in every human cancer cell line, the whole NCI 60. And it did clinical trials in solid tumors, and it just wasn't successful. And the reason boiled down to the fact that the therapeutic window was just too narrow. And so the drug can work in a liquid cancer where, as soon as it's administered, basically immediately exposed to all of those circulating tumor cells. Not to mention, it is maybe about a factor of 5 to 10 more potent than multiple myeloma, which is a good mechanistic reason. Their characteristic is overexpression of antibodies, so they can't degrade those mechanisms down. But what this meant to us is that this drug, it's got a potent mechanism of action for single-digit animal or human solid tumor cell lines. But it just doesn't get to solid tumors. And the big reason why is it turns out that red blood cells, which are everywhere in your blood, have high proteasome concentration. And actually, over 90% of every molecule of melting that's introduced into the body actually goes to red blood cells, basically binding its stuff. So it's really low bioavailability, poor biodistribution, and you just don't get enough drug to solve it. And you can't go more because you have about a 1 log per week. And so we thought, well, what if we've conjugated this onto our Bottlebrush and we can target selectively cells that are expressing epitopes with antibody-mediated cell uptake, maybe we can overcome this and introduce proteasome inhibition, a new mechanism of action for ADC base. And we were happy to see that indeed, if we look at the concentration of our construct, and particularly the bortezomib in blood over time, and let me explain this a little bit. The green circles here are whole blood, and the white are it's just serum, sort of separate. And the idea here is that if the drug all goes into red blood cells, if you separate them, what you'll see is that there's none of the serum. right? And in fact, we see that these points basically overlap, meaning we're keeping the drug out of those red blood cells, which makes sense because it's conjugated to our conjugate. By contrast, and this is actually over several weeks, bortezomib on its own, 2 hours, 2 days post-dose is basically unfair. All right. So with that in hand, we have tested the applicability of this proteasome inhibitor antibody Bottlebrush conjugate in a couple, well, actually several different models. I'm just going to show data from 2 of them. Actually, 1 model, 2 sets of experiments today. First is we're using a model here called HCC70, which is a breast cancer model that is actually technically classified as triple negative. It doesn't express HER2, but its expression is not 0. It's very low. And we have this hypothesis that in low antigen expressing cells, having a higher DAR would also confer a benefit, because for every antibody conjugate binding event, you bring more drugs into that set. Right? So and also, just to be frank, like in HER2 is so good in HER2. If we want to take this further, like we can show efficacy in models where HER2. And here, what I'm showing is a comparison of TDXD, which isn't HER2, it's the biosimilar that's commercially available, given that it's dosed clinically can be okay, and ADC IgG1, which is the control, not binding anything. You see it doesn't work so well compared to control. But if we dose, and I'm giving the doses here in 3 different versions to give you all the information. So if you dose by the antibody, so in HER2 10 mg per kg, we have 0.5, 1.73, 15. This is the ADC total dose, and this is the payload. But long story short, we can see a nice dose response. We're even at this really low dose, 0.5 mg antibody, we're getting performance of par in HER2, which is the clinical gold standard, and it's using a payload exatecan, which is about 10 times more potent than molecule. But if we go a little bit higher dose, and these are well-tolerated doses, we get better performance, and at this highest dose, we see significant tumor regression, although not completely compared to this study. And so the last bit of data I'll show you is one that I think highlights the importance of this DAR value. And many people I talk to are like, well, do you really know if you need to have the DAR? In this case, by the way, the DAR is 60. Yeah, I realize it doesn't say the couple of you really need that? What about DAR? And so again, the hypothesis is that higher DAR in a low antigen expressing tumor would potentially confer a benefit because there's simply more drugs per antibody. And here we did an experiment. I don't know that you could do this with any other system. Please tell me if I'm wrong. But what we were able to do here is we made 3 different ADCs, all with bortezomib as the payload, where we only changed the DAR, or 10, or 30, or 60. But importantly, so you know that the DAR is related to the length of that molecule. So if we go 60 to 30 to 10, we're significantly changing the length, right? So that's changing 2 variables. I don't like that. So instead, what we did here was to make a DAR 10, we co-polymerize a monomer with the bortezomib and another PEG monomer that doesn't have a drug. So we still have a length of 60, but only 10 drugs. So we now have so DAR 10, DAR 30, DAR 60 all have exactly the same length, exactly the same size, right? All the things that get the only different by the DAR. And here, what you see is that compared again to TDXD, which is the HER2, we also use lower doses in this study simply because we wanted to try to amplify differences between DAR. And what you see is that the DAR 60 significantly outperformed both the DAR 30, the DAR 10, and in HER2 in this study. Okay, so with that, I'm running out of time, so I'm just going to end with a quick plug. So a couple of years ago, I was really happy that 2 of my former PhD students, Hung, who was shown on the previous slide, and Yvonne, I'm not highlighted in this talk today, but a former student in the lab, we co-founded a company called Window Therapeutics. Catchy, right? Here's a picture of the team as of maybe like 6 months ago or so. We've got a great team, and if anybody's interested in learning more about Window, feel free to ask me, especially if you have $1 million you want to give me. And with that, I am very happy to take any questions. Thank you.